Biology Station by Pyruvate.
Most of a eukaryotic cell's ATP synthesis occurs in mitochondria. A mitochondrion is a sac within a sac. The inner sac is folded, increasing surface area for ATP synthesis. Between the inner and outer membranes is the intermembrane space, a reservoir for hydrogen ions used for synthesizing ATP from ADP. The inner chamber, known as the matrix, is a soup of enzymes that dismantle fuel molecules in the Krebs cycle. The knobs are where ATP is synthesized. Simply put, a mitochondrion is an energy transformer, where energy in fuel molecules such as pyruvic acid is transferred to ATP. Carbon dioxide, the carbon end product of cellular respiration, diffuses out, leaving the cell through the plasma membrane.
In this video, we are going to look at glycolysis. Glycolysis is the process of breaking down a glucose molecule into pyruvate and gaining a couple of ATP and NADH in the process. Glycolysis begins with the molecule glucose. The enzyme that begins this process is called hexokinase, or glucokinase in the liver. A kinase is an enzyme which adds phosphate groups onto things by taking them off an ATP. And that's exactly what happens here. An ATP donates a phosphate to the glucose and creates this molecule, glucose 6-phosphate. It seems odd to use ATP in this process because we are trying to create ATP, but we'll see that we get all this ATP back plus more once the process is complete. The glucose 6-phosphate is acted upon by phosphohexose isomerase. Isomerases are enzymes which rearrange the structure of the substrate without changing the molecular formula. We can see that the phosphohexose isomerase rearranges the glucose 6-phosphate into a fructose 6-phosphate molecule. Now we meet another kinase, phosphofructokinase 1. This enzyme attaches another phosphate group to our molecule, creating fructose 1,6-bisphosphate. Sorry, my mistake, that should read fructose 1,6-bisphosphate, not bisphosphonate. Anyway, moving on, the molecule is then split in half by fructose bisphosphate aldolase. An aldolase is an enzyme that creates or breaks carbon-to-carbon bonds. which makes it the enzyme of choice to split the fructose 1,6-bisphosphate molecule into these two molecules, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. The next step is conducted by another isomerase, the triose phosphate isomerase, and this converts the dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. The next enzyme we meet is glyceraldehyde 3-phosphate dehydrogenase. The dehydrogenases move a hydride ion, that is H-, onto an electron acceptor such as NAD+ or FAD. This is what happens here, creating 2 NADH. There are 2 molecules of NADH created because one glucose molecule created 2 glyceraldehyde 3-phosphate, one from the aldolase reaction and one from the triose phosphate isomerase reaction. The hydride group on the molecule is replaced by a phosphate group supplied from inorganic phosphate, not from ATP. It is the reduction of NAD+ into NADH which provides the energy to bind inorganic phosphate. This gives us the molecule 1,3-bisphosphoglycerate. Here the molecule meets another kinase, phosphoglycerate kinase. We saw above that kinases can giveth phosphate, but they can also taketh phosphate away. And that's exactly what happens here. 2 ATP are produced by taking phosphate from the 1,3-bisphosphoglycerate molecules. This produces the molecule 3-phosphoglycerate. The enzyme phosphoglycerate mutase is essentially an isomerase, so it creates the isomer 2-phosphoglycerate from 3-phosphoglycerate. Enolase is the next enzyme and it produces a double bond by removing the hydroxyl group. This leaves us with phosphoenol pyruvate. The final enzyme of glycolysis is pyruvate kinase. And this kinase is also going to remove phosphate groups from the molecule, attach them to an ADP and create ATP. Again, 2 of these are created because 2 phosphoenol pyruvates will be created for every glucose molecule. This finally leaves us with pyruvate, which will be the substrate for the Tca cycle. Now for your reference, I shall quickly number these steps, and you can see that there are 10 steps in total. Steps 1 to 5 are often referred to as the investment phase because they use up ATP. Steps 6 to 10 are known as the payoff phase because we get back our initial 2 ATP investment plus 2 additional ATP and 2 NADH, which will later become even more ATP. And that's glycolysis. In the next tutorial, we will be having a look at the fate of pyruvate. The primary substrate for the Tca cycle is pyruvate. Fatty acids and amino acids can also be used, but we're not going to discuss them in this tutorial. Pyruvate is produced by glycolysis. And the first thing that happens to pyruvate is that it gets converted into acetyl coenzyme A, more commonly known as acetyl-CoA. This conversion happens via the action of three enzymes, collectively known as the pyruvate dehydrogenase complex. When you see the word complex after an enzyme, it usually means there are multiple enzymes involved, but they are functionally considered as one. As we saw in the glycolysis tutorial, dehydrogenases are the enzymes which produce molecules like NADH and FADH2. Accordingly, the pyruvate dehydrogenase complex produces an NADH molecule. Now I've drawn the acetyl-CoA as two balls of carbon. This is because acetyl-CoA is a 2-carbon molecule. In this tutorial, we're going to pay special attention to the carbon balance throughout the Tca cycle. Hence, it is easier to illustrate the molecules involved in this way. Note that acetyl-CoA has one less carbon than pyruvate, which is a 3-carbon molecule. The extra carbon is lost as carbon dioxide as a byproduct of the conversion to acetyl-CoA. We'll look at the molecules involved first, and then we'll discuss the enzymes involved. The Tca cycle begins when a 4-carbon molecule known as oxaloacetate combines with acetyl-CoA to produce a 6-carbon molecule known as citrate. Citrate becomes the 6-carbon molecule cis-aconitate, and then cis-aconitate becomes the 6-carbon molecule isocitrate. In the next step, we lose one of these carbons as carbon dioxide, and we also produce another NADH. Thus we are left with a 5-carbon molecule known as alpha-ketoglutarate. The same thing happens again. We lose a carbon as carbon dioxide and gain an NADH. Hence we are left with only 4 carbons in our main molecule, which is now called succinyl-CoA. Succinyl-CoA is then converted to succinate, and a GTP is produced in the process. GTP is essentially an equivalent of ATP. Succinate is then converted to fumarate, and an FADH2 is produced in the process. Fumarate becomes malate, and malate completes the cycle and becomes oxaloacetate, producing the final NADH in the process. The oxaloacetate is then available to combine with any available acetyl-CoA and begin the whole process again. Now let's take a look at the enzymes involved. Citrate synthase predictably synthesizes citrate from oxaloacetate and acetyl-CoA. Then, the enzyme aconitase produces cis-aconitate and then the conversion back to isocitrate, which is a structural isomer of citrate. Again we see dehydrogenases in action, with isocitrate dehydrogenase producing the NADH in the isocitrate to alpha-ketoglutarate conversion. And the alpha-ketoglutarate dehydrogenase complex also producing NADH in the conversion to succinyl-CoA. Succinyl-CoA synthetase catalyzes the conversion of succinyl-CoA to succinate. Then yet another dehydrogenase, known as succinate dehydrogenase, produces the high-energy molecule FADH2. Fumarase converts fumarate to malate, and then the final enzyme is another dehydrogenase, malate dehydrogenase, producing NADH and converting malate to oxaloacetate. And that's an overview of the Tca. This is the fifth and final video in the cellular respiration series. In this video, we're going to look at oxidative phosphorylation, which is also known as the electron transport chain. You may remember that a mitochondrion looks a little like this. It has an outer membrane and an inner membrane. And we're going to take a close-up look at the inner mitochondrial membrane. So here is the membrane, and within it are a bunch of protein complexes. They are known as complex 1, or NADH dehydrogenase, complex 2, or succinate dehydrogenase, complex 3, or cytochrome C reductase, and complex 4, or cytochrome C oxidase. These are the major protein complexes of the electron transport chain. The other factors involved in the electron transport chain are coenzyme Q10 and the cytochrome C complex. The other large protein complex involved here is called ATP synthase, and as the name suggests, it is the complex that makes ATP. So the process begins with NADH, which was produced by glycolysis, beta oxidation, and the Tca cycle. This NADH is oxidized to NAD+ and donates 2 electrons to the electron transport chain. These 2 electrons get passed between the complexes like this, and are finally accepted by oxygen, and with a couple of protons, this produces water. This oxygen is supplied by breathing air from our atmosphere, and to provide this is the reason that all animals need to breathe. Now, as the electrons are passed between the complexes, they pump protons out of the mitochondrial matrix into the intermembrane space, like this. Complex 1 pumps out 4 protons, complex 3 pumps out 4 protons, and complex 4 pumps out 2 protons. Therefore, in total, one NADH pumps 10 protons across the membrane. A similar process happens when FADH2 is used as the electron donor. 2 electrons get donated to complex 2, which in turn passes them to coenzyme Q10 and onward down the electron transport chain. Because complex 1 is not involved when FADH2 is the electron donor, only 6 protons get pumped across the membrane. Finally, these protons then flow back down their electrochemical gradient through the ATP synthetase. For every 4 protons that flow through the ATP synthetase, a single ATP is produced. I should just say that different textbooks will quote different numbers here. I've quoted the number from Leninger's Principles of Biochemistry, 5th edition. So if you do the math, it turns out that one NADH will produce 2.5 ATP, and one FADH2 will produce 1.5 ATP. One important fuel for the cell is glucose. The body acquires glucose by breaking down carbohydrates in the diet into their simplest form, which is often glucose. Glucose is converted into this molecule, pyruvate, through a series of steps known as glycolysis. We'll look at those steps in a later tutorial. The process of glycolysis produces two of these ATP molecules. It also produces two NADH molecules. These NADH molecules will be converted into ATP later. The pyruvate molecule then undergoes another series of reactions known as the Tricarboxylic acid cycle, more commonly known as the Tca cycle, but it's also known as the citric acid cycle and the Krebs cycle. The Tca cycle creates a guanosine triphosphate, which is essentially an ATP, an FADH2, which will get turned into ATP later, and 3 NADH, which will also get turned into ATP later. The Tca cycle also produces carbon dioxide as a byproduct. Now, pyruvate isn't the only substrate for the Tca cycle. Amino acids can be used in the Tca cycle, and so can fatty acids, via a process called beta oxidation. We'll look at the Tca cycle and beta oxidation in later tutorials. Note that when glucose is the substrate, one glucose molecule creates two pyruvate molecules, and this leads to the production of twice the amount of GTP, FADH2, and NADH produced by the Tca cycle. The FADH2 and NADH molecules are then used to create ATP, which, as I said before, is the energy of the cell. This process happens via the electron transport chain within the mitochondria of the cell, and this is a part of the process that requires oxygen. The reasons that humans require oxygen is to supply it to the electron transport chain and create ATP. All in all, one molecule of glucose can create a maximum of 38 ATP. However, this is not really an accurate number, as some of the energy is used moving all these molecules into different parts of the cell, so the actual number depends on how you decide to count the ATP. And that's an overview of cellular respiration. In the next video, we'll have a look at the